Hi, so this is a short video about modelling disorder in crystals. We're going to run through an example. So first, some quick definitions. So disorder occurs when unit cells in the crystal contain different contents. So it might be different element types, it might just be different conformations of molecules. Ideally, for us to model it, this disorder is uncorrelated. So that means the configuration of one unit cell doesn't depend on its neighbour. So we've got a picture here of a disordered crystal with a diatomic molecule of some sort, and you can see in 3 out of the 9 unit cells it has a different conformation. Now, what we need to do in our crystallographic software is to construct the average unit cell in the crystal. So in this case, we would want kind of an overlay of these two molecules, and we'd want 1/3 occupancy of the molecule where there's pointing upwards to the right, and we'd want 2/3 occupancy of the molecule that's pointing down to the left. So you can imagine all of those compressed into one unit cell, sitting on top of each other, each with a partial occupancy. Now, crucially, those occupancies can't add up to more than one, so we're still modelling a real crystal. So we know that it has to be physically meaningful, and the total occupancy of molecules in one unit cell can't be more than one. So we can use that as a constraint. Okay, so let's run through the example. We're going to start crystals here, and the best place to start it is in the demo folder. Click OK, and actually let's choose Workshop Demo from the Help menu, Proceed, and it gives us a list of demo structures to choose from. The one we're going to look at in this video is this one, Zurich Disorder. Open up the workshop structure. Okay, and then this part is common to all the videos, so if you have any, if I skip over things a bit fast, go back and watch the video of the standard routine structure, and it goes through it a bit more slowly and introduces you to the interface. So import the data. All the data is in this file called import.cif. In this particular import format, we have to click here on the command line and accept the default options of this program that's running inside crystals here. So it wants to suggest a space group of P21 upon C. If we went back and looked at the absences, we would agree with that, and it suggests, based on our formula that we've given it in the input, that there might be 4 of those molecules in the unit cell. Okay, now it suggests reading in the HKL file, which has been extracted from that import.cif. At this stage, we would have to remember what the size of the crystal was, what shape it was, what colour it was, the pressure we did the experiment, and the temperature we did the experiment. This stuff just gets stored away for later. It doesn't affect our analysis at all. Okay, so initial analysis of the data. We've covered these in the previous video. We can skip through these. Nothing too weird going on here. And all of these numbers look okay. So the volume per non-hydrogen atom seems to be about 18, and our data to parameter ratio looks good. Right, solve the structure. So we'll run superflip. It runs away in the background. Sorry about the flickering screen. P21 upon C is confirmed by the charge flipping analysis, and here is our structure. It looks good so far. So if you went and read the file that comes in this folder, you'd recognize this structure as the one that we were probably looking for. And let's dive in with some refinement straight away. Positions and isotropic temperature factors. Okay, so far it kind of looks okay. This carbon atom here looks a bit big, so your initial instincts might be to think, well, maybe that's the wrong atom type, maybe that's a... No, actually I don't know, there's probably no reasonable explanation for that. Let's try anisotropic refinement. Okay, oh, look at that. So this is a classic symptom of conformational disorder of some sort, where an atom is moving between one side of a plane and another, or it's waving around, so it's telling us that in some unit cells the carbon atom is up here. In other unit cells, it's down here. And those are actually, if you look carefully, those are actually enantiomers of each other, because this is an sp3 carbon, and the hydrogen will be pointing in different directions. So, how do we deal with it? We need to construct an average model of that disorder. So in crystals there's a facility here, split atom, we can split it into two atoms, and it breaks that ellipse. The foci of the ellipse, it places atoms. So we've got, instead of C5, we've now got C51 and C50, and this little label here is telling us that the occupancy of both of those is a half, so they correspond to half the unit cells having this carbon in and half of them having that carbon in. The other atoms are all full occupancy, so they've got the same configuration. Okay. What we can do in crystals as well to help it along with the refinement, we can actually assign these atoms into different parts. So we've got columns here where we define an assembly and a group, and in crystals notation we combine these two numbers together. So we do 1000 times the assembly, which is the general area of disorder, so this area of the molecule here might be assembly 1, and we split it into two groups. So this would be group 1 and this would be group 2. And in our crystals notation then, set the part number, this would be 1001, so it's 1000 times the assembly number plus the group number, and this one would be 1002. And you can see crystals helpfully colours the different groups, the bonds to the atoms in different groups in different colours, so you can kind of see what's going on. Right, let's go back to refinement and click OK. And crystals refines away. And what I should have pointed out here is, let's do it again. In this dialog, there's a new box has appeared. It's detected there's some disordered assemblies, and it says it's going to refine the site occupancy factors of the assemblies, keeping the sum at 1. So those occupancies are allowed to change, and they should therefore reflect what the distribution of disordered molecules is in the crystal. So we're currently at 0.54 and 0.456, but they add up to 1. Okay, let's carry on with the guide. No extinction required, and it's a little bit stuck here. The R factor is 12%, so I'm going to suggest that we add the hydrogen. And now it gets a bit more complicated, but crystals takes care of it all for us. So onto this carbon 51 is added a hydrogen with giving it the same occupancy, and the same here on carbon 50. It's the neighbouring atoms where it gets a bit more complicated, so this vector into this carbon here is different depending on which of the two conformations you're in, and therefore the distribution of hydrogens on this sp3 carbon, this methyl at the end, are going to be different as well, and so they also end up in different parts with different occupancies. But all of these things are linked together. There's only one parameter being refined which defines the occupancy of the whole thing. Similarly on this carbon up here, we've got two hydrogens on an sp3. Ignore the pink atoms for now, it's just the white ones it's placed, and the angles vary depending on whether the neighbouring atom is the carbon 50 or carbon 51. Okay, all hydrogens have been found, done. Now it's doing some refinement, nothing's really moving, and it will close down in a second. Right, a bit more refinement, and that's the end. Structure validation, all checks passed successfully, and then we would carry on to check this on the web if we wanted to check it in a bit more detail. So over here is our average model of the crystal structure. It's made up of two enantiomers, and for some reason, maybe because the bulky part of the ring over here is common to both of them, they've packed in a disordered manner instead of normally these things would pack with a centre of inversion between them. So an interesting little structure, and that's how you deal with it in crystals.